So the next topic is protoplast isolation and fusion. How we can get again interspecific hybrids or development of hybrids, right? Cytoplasmic hybrids. So you might have already seen or heard of this tomato or tomato and a potato, where in a single plant, both are of the Solanum species. Like Solanum species, where you have tomato on the shoot and potato in the root system, right? So this can be obtained by this interspecific process. Solanum lycopersicum and the Solanum tuberosum. Solanum tuberosum is the potato, and Solanum lycopersicum will be this tomato, where we can get interspecific hybrids or somatic hybrids. So the protoplast. So what a protoplast? So that will be in a 1 mark or 2 mark question. Right. So the protoplast refers to all the components of the plant cell devoid of the cell wall. So if we remove the cell wall, also in fungi, we can remove the cell wall and we can obtain the protoplast. So just the plasma membrane surrounded with the organelles, surrounding the organelles, it will be called as the protoplast. So these were first isolated using a mechanical isolation, and in most of those cases, the yield of protoplast was very small or not uniform, and the survival was also very low. And nowadays, we isolate protoplast by enzymatic method, also by enzymatic digestion of those cell walls. Right. So the advantages of protoplast culture is it's the simplicity and ease of control of a single cell to manipulate the entire genetic material. So we can manipulate, take out the organelles or the genome, and then put in other components inside the cell. So that is much more easier, which is highly impossible task in complex eukaryotic system like the plant cell, where you have the cell wall and where you cannot inject anything inside. So in eukaryotes, the distance hybridization or wild hybridization is also which is not possible. So this is hampered by the sexual incompatibility barriers. So you cannot develop any embryos from wild hybridization or distant hybridization. So this protoplast the fusion of those somatic cells could be effective to make this parasexual process, interspecific process at the cell level between two distant relative plants. And further, this is also possible to transfer foreign DNA or even entire organelles, cell organelles. So there have been instances of transferring chloroplast or mitochondria from one species into another species. So entire organelles can also be transferred, or bacterial genome or the virus genetic material, usually for this genetic manipulation of this crop species. So the methods of protoplast isolation. So this can be in a 5-mark question, right? So we have this mechanical and also the enzymatic isolation methods of protoplast. So the mechanical isolation. So what you take the entire the leaf, entire leaf blade, and put it onto an isotonic solution. So here, when we look at the leaf cells, no, usually we get it from the parenchymatous cells. So parenchyma cells or large cells inside the leaf. So where you have all this towards the cell wall, the plasma membrane will be attached to the cell wall, almost attached to the cell wall. So this will be very difficult to release. So put it in an isotonic solution, isotonic for plasmolysis, right? So when you put it in an isotonic solution, the water will go out so that the plasma membrane will detach from the cell walls and then form a sphere, right? So by plasmolysis, we get the plasma membrane detached from the cell wall and form a sphere. So under a microscope or just simply just cut, give vertical cuts to this leaf, right? So anywhere, so just randomly, there will be cut in the cell wall, and this spherical, the protoplast will be released outside, right? So you cut it, and then where there is cell wall cuts, not in all the cells. So in few cells, there will be cut, and from that, those protoplasts will be released outside. So these released protoplasts will be taken up in an isotonic solution, right? To give the osmolarity, right? Here, osmolarity in the sense because this is inside, this is under pressure, right? All those plasma membrane inside the cell is under pressure, right? So when we take it outside, there will be lots of pressure inside the cell, so that when it comes out into the atmosphere, that will start to burst. So for that, we need to, this needs to be released in an isotonic solution. So the first mechanical isolation was described by Glecker, and the cells are first plasmolyzed, causing the protoplast to shrink and reduce away from the cell wall, and the cut is made using a scalpel, and some of the cell walls will be cut without damage to the protoplast, and when the tissue pieces are deplasmolized, the protoplast within the damaged cell will come out, will swell, and be squeezed out into the bath culture medium. So the main advantage of this physical or mechanical isolation method, is that the unknown effects of using enzymes. So when we are studying the activity of hormones, some of those enzymes, which is isolated mostly from the microorganism, may also contain some hormones. So that can be unknown side effects of the enzyme mixture can be eliminated. That's the only the advantage of that. The most disadvantages is that there is very less amount of protoplast being produced, and that is not all the protoplasts will not be of the same size or normal sized. So the other method is the enzymatic isolation. So enzymatic isolation, you need to remember enzymatic isolation discovered by Cocking, right? Demonstrate the possibility of enzymatic digestion of the cell walls. So they are using concentrated solution of cellulase isolated from the fungus, and also we can also use the takabe, used many of those commercially prepared enzymes. Like these are all those commercially available enzymes that we normally use for protoplast isolation. Say like cellulase for dissolving the cellulose. Say like the enzymes, what we need, like for a cell, for a plant cell, you have this cellulose, hemicellulose, and the pectin, right? So these three materials needs to be digested. So for cellulose digestion, you need cellulase. For hemicellulase, and for pectin digestion, you need pectinase. Right? So these are all the three enzymes that is needed to digest a plant cell wall. So this cellulase R10 from isolated from Trichoderma viride, then hemicellulases from a Rhizopus maserozyme. So this enzyme has both pectinolytic and also cellulolytic activity, this maserozyme. And you have pectinase, pectolase. So all this isolated from different fungi. So these are all commercially available enzymes that is used for digestion of the plant cell wall. So the isolation using enzymatic digestion is the sequential method and simultaneous method. Sequential, as the name suggests, sequentially. Sequential method is a two-step method where the first one using isolated, first treating with the macerozyme, which is a pectinase. Right. So first, when you look at this structure of the cell wall, you have this cellulose microfibrils. So where you have this hemicellulose, side branches. So all this are being like cemented together by the pectin. So first remove this pectin using a pectinase, maserozyme in 13% mannitol. So 13% mannitol, this will be the osmoticum, right? So osmoticum, why do we need osmoticum? To reduce the pressure inside the protoplast. So because when protoplast comes out of the cell wall, it will tend to burst. So to prevent that, you need to have this osmoticum. Osmoticum will be some sugar alcohols, 13% mannitol. So the isolated cells are obtained and incubated and then purified. And then again, finally, after removal of this pectin, remove all those cellulose and hemicelluloses by using cellulases. Right, this will be the two-step method. First, remove the pectin and then remove the celluloses. Right, so this is cumbersome method. All right, first do it, then purify, wash it, and then again use cellulose. So this is a cumbersome method, and we can do both this enzyme treatments simultaneously, which is called as the simultaneous method, right? Or direct method or one-step method, where we use simultaneously both the enzymes. 0.5% of pectinase and 0.3% of cellulase in 13% sorbitol or mannitol in a pH 5.4. These are mixed and used simultaneously. And the cells are incubated in the mixture for 15 to 18 hours at 25 degrees centigrade. So where the entire cell wall will be digested and the protoplast will be released inside the media. So the factors influencing the isolation of protoplast. So isolation and viable and cultural protoplast is affected by the source material. So which we select the leaves. At least it should not be too mature or too young. Right, I use normally matured young leaf. The petals, petioles, shoot apices, microspore, pollen, callus. Any cell can be used to isolate the protoplast. The pre-enzyme treatment. Right. So how do we plasmolyze? Plasmolyze this also. The pre-enzyme treatment also affects how much of protoplast is released inside the media, and the enzyme treatment. What type of enzymes we use on the concentration, and finally the osmoticum. What we use, the type of osmoticum and the time we use the osmoticum also plays how much the quantity of protoplast we get from that. So the enzyme pre-treatments, what we do to the explants are subject to the first, the surface sterilization. So a surface sterilize using a Zephyra or any other surface sterilant what we use, followed by 10% ethanol for 5 minutes, and the pre-plasmolysis conditions. So to minimize the effect of the enzymes on protoplast yield, so these are placed, the leaves are placed in pre-plasmolysing solution for 1 hour prior to putting on this enzyme. So this also reduces the amount of enzyme uptake during the protoplast concentration or isolation. So this shrinking of this protoplast from the cell wall also seals the leakage of the cell contents due to sealing of the plasmodesmata. So this pre-plasmolysing solution should be of the same osmolarity as that of the enzyme solution. So whatever we use, like 13% mannitol, what we use, it should be of the same and may contain other salts of the culture media plus an osmotic stabilizer like DMSO. All this can be used as a pre-plasmolytic solution. So the next is the pre-digestion treatment that facilitate the penetration of the enzyme solution into the intercellular spaces. So mostly we can also peel the lower epidermis. So the lower epidermis contains all those stomata and all other say like trichomes. So those lower epidermis can also be peeled. So for the easier uptake of the enzyme inside the cells, and float the stripped pieces of leaf on the enzyme solution so that the peeled surface is in contact with the enzyme solution. And where peeling is a problem in very small leaves where we cannot peel the undersurface, so the cut the leaf tissues into strips and then float onto this enzyme medium. So this can take the leaf and then cut it into strips, into small strips, and then float it onto the media. So where there can be a pre-plasmolysis, right? So those entire protoplasm will come away from the cell wall and then form a circular shape. And then when it is being digested, you get an entire protoplast devoid of the cell wall. So the enzyme treatment, the actual enzyme treatment is dependent on the nature of the enzyme and its concentration. So what type of enzyme what we use. So again, this needs to be standardized for because each of those leaf cells. So like hardy plants will have more of pectin, like rice. Rice will have more of pectin, or say some other plants, say like vegetable plants, like the greens, they will have less of pectin. Right. So depending upon the leaf type, we need to standardize the amount of enzyme what we need to use. So the three types of enzymes can be used: cellulases, pectinases, cellulase to remove the cellulose, pectinase to digest the pectin, and hemicellulose to digest the hemicelluloses. cellulose medulla, and pectinase to degrade the middle lamella, the cementing layer. So some tissues may require also hemicellulose in addition to cellulase and pectinase. And usually we use the crude commercial enzymes, which can also contain nucleases, proteases as impurities, which may be harmful to the protoplast viability, but that cannot be overcome. So most of this commercial enzyme can also contain all this. But if we are going for, say, like transgenes, injecting DNA inside, we need to use purified forms of enzymes, not commercial enzymes. Next, the successful release of protoplast also depends on the pH, temperature, the treatment time duration, the concentration, and the volume of enzymes being used. And also the use of osmoticum, what type of osmoticum, and the timing, the concentration. This also influence how much protoplast we get. So the protoplast released from the cell wall will burst when they are transferred to the culture media because of the differences in pressure. So because of the entry of solutions. So when the cells are in contact, the pressure exerted by the cell wall prevents bursting of the protoplast. So this can be prevented by maintaining appropriate osmotic pressure inside the protoplast. So various sugar alcohols are used as a osmotic, like the mannitol, sorbitol, and some sugars, sucrose, glucose. Mostly we will be using the mannitol and sorbitol, either in separately or in combination. This being used normally. Usually we use 13% mannitol or sorbitol. The purification, protoplast purification. So this the solution. So the solution what we get used to digest the cell that may contain undigested cell, the broken cells, vascular elements, protoplast, other parts of cell, along with the healthy protoplast, intact healthy protoplast. So these are purified using filtration. So the first steps is the filtration using 50 to 100 millimole pore size, just using a muslin cloth. Just using a muslin cloth, purify this purification by filtration, and then centrifugation at 100 RPM, not too much. If this too much RPM, you will break the protoplast. So just 100 RPM for 7 to 10 minutes, it precipitates the intact protoplast, and the intact protoplast and the floating protoplast can also be siphoned off using a passive pipette. All other debris can also be removed using a passive pipette, and then washing with sorbitol solution. And after washing, those protoplasts needs to be tested for viability. So viability testing of protoplast using the FDA. Right? So FDA fluorescent diacetate. FDA is a substrate for esterases, right? So where do we use esterases? Esterases, esterase peroxidase. So these are all used to remove the hydrogen peroxide that is produced during respiration. Right. So whatever the cells are respiring will produce hydrogen peroxide. Right. So when we apply a substrate like the FDA, fluorescent diacetate, so this will cleave the hydrogen peroxide and then give a color. Like the fluorescent diacetate when applied to this living cell. So because of this presence of this esterase in the living cell, so this will cleave the fluorescent diacetate into fluorescent, which will fluoresce green color. So all the live cell will be green fluorescing. Right, all other dead cell will not have any color. So by this we can know how much of the live cells are present. So this gives, which is fluorescent, which is released and is not freely permeable across the plasma membrane. So this will be taken inside the cell, and the cell will be giving green fluorescence when observed under UV light, and which is usually used in a concentration of 0.01%. Or Evans blue staining, which is used for dead cell staining, Evans blue, so which is taken up inside the cell. So the dead cell will have permeable cell wall or the membrane. So Evans 0.025% solution of Evans blue, the damaged cell take up the stain, but the viable cell excluded and remain unstained. So by this, we can see the dead cells. Or phenolsafranin, again, stain the dead cells. So also the phenolsafranin at 0.1% also used to detect the dead protoplast, and the live cells remain unstained. Or just for phase contrast microscopy, so we can see the cytoplasmic streaming. So when you look at the protoplast or the leaf cells, we can see those chloroplast or cytoplasmic moving inside the cells. This can also show how much of the cells are viable. So this is the FDA staining. So all those live cells, they will be green color fluorescing, and all other dead cells will not show any fluorescence. Right? By this, we can see how much percentage of cell is live, and we can also count the number of cells that is live. So we need to count the number of cells before plating or culturing, right? So for successful culture, we need to determine the number of protoplast that is used. So we cannot just use one or two protoplasts for culturing. So there need to be a minimum number of protoplasts that is used for culturing, which is called as the plating density, right? The minimum number of protoplast or cells that is needed for culturing is called as the plating density. So for tobacco, we need to have at least 5,000 to 1 lakh protoplast per ml of the medium. So this will be the optimum. 1 lakh the protoplast per ml of medium is the optimum plating density for culturing. Now, tobacco propolis. These need to be inoculated in the medium at a minimum plate density, which is called as the minimum plating density. In other words, if for the minimum, if there is not the minimum density, the protoplast growth of protoplast will be affected, and the concentration of protoplast in the given preparation can also be counted using the hemocytometer, right, which is called as the Neboy's hemocytometer. So this cell counter is also used for platelet or any other cell counting, bacterial cell counting or blood cell counting. So that counter hemocytometer can be used to count the number of protoplasts per ml or per microliter of the medium. So the plant regeneration, the advantages or the process in generating the plant from the protoplast is that it will first form the cell wall. After we purify and put it onto the media, it will start to first isolate the protoplast and then determine the longevity or the survival of those protoplasts. So all those FDA taken protoplasts will be green, and all other dead will be red, and the chloroplast will fluoresce red under UV light. So these are all dead protoplast under the green or live protoplast. So after plating onto the media, they will form this microcalli. So this microcalli will form the callus, actual callus, and then form the regeneration of the shoots. So first, after we take up the protoplast, it will form the cell wall and will start to divide. It forms the cell wall and the cell division, and then it forms the microcalli and the regeneration of plantlet from the microcalli, right? So the protoplast culture from ovule derived callus of sweet orange. This is another example where you isolate the protoplast, develop calli, and get the plantlets from that. The rice, orange. So this is from rice. So you get a freshly isolated protoplast. So the cells start to divide after forming cell wall, and this protoplast can be embedded into agarose, right? Very thin layers of agarose, which can also be floated, blocks floating onto the culture media, right? So the culture or the callus will start to divide, and then the callus will form the shoots, right? So from this, we can isolate from the protoplast. We can isolate the entire plantlets. This is from the banana, where the protoplast can form somatic embryos. And from somatic embryos, we can regenerate the plantlets from banana. So the applications of protoplast culture. So protoplast can be induced or fused to produce hybrid plantlets. which is not possible using conventional plant breeding because of incompatibility, or it is capable of taking the foreign materials, say like it can be mitochondria, DNA, plasmids, bacterial, viral genome. Entire thing can be taken inside or injected inside the protoplast. The entire mitochondria can be injected inside the protoplast. Or the protoplast can be used to study cell wall synthesis and also deposition, right? How the cell wall is being formed. Because from the first class we have seen, we still don't know how the cellulose being synthesized from the plants. So still we are studying how the cell wall is being synthesized and also the transport of metabolites from the cell wall. And the protoplast can also be used to study single cell system. How a single cell functions on its own can also be used using the protoplast. So the other next topic is the protoplast fusion. How we can fuse the protoplast and get the hybrids, cytoplasmic hybrids. Protoplast methods of fusion, methods of selection of those fusions, and the application of this protoplast fusion. So this normally for in agricultural crops, sexual hybridization has been utilized to give special characters. So the limitation of the sexual hybridization is that it can be used only within the species and their wild relatives. Because of many barriers, it is not possible within interspecies or intergeneric. It is not possible. So now we can use hybridization of a somatic cell. Somatic cell hybridization, also called as parasexual hybridization, can be done using protoplast fusion. So normally, if we take this is again discovered by Withers and Cocking. So you should not forget this name, right? Again, Withers and Cocking. So take a protoplast, and when we bring two protoplasts close together in a very close proximity, right? Just like pushing inside. So just like bubbles. So when we blow the bubbles, like soap bubbles, so when we have two soap bubbles close enough, then it can fuse to form a larger bubble, right? Or if you don't have much of conditions, then the soap bubble will burst, right? So we need to maintain the condition, and when we bring the two bubbles together, two protoplasts together, they can fuse together, right? So this can be spontaneous. The fusing can be spontaneous, can also be induced, right? So this, when we bring in two protoplasts together, the two protoplasts can fuse together to form an individual protoplast. Protoplast technique is in vitro technique to have genetic recombination. So you can have the entire genomic fusion. Genomic is it can be nuclear fusion or the cytoplasmic fusion alone, right? Either the nuclear or cytoplasmic fusion. So there is no problem of pre-zygotic barriers. So there is no barriers that can prevent this. Result is the production of asymmetric hybrids. Asymmetric is it is not equal fusion. So the nucleus may or may not fuse, or the cytoplasm may or may not fuse. We will get nucleus from one plant and the cytoplasm from both the protoplasts, right? So these are all asymmetric, right? Asymmetric hybrids. So no problem of genetic barriers, and also known as somatic hybridization or parasexual hybridization. So methods of fusion. The fusion can be first spontaneous fusion that is normally occurs within a few hours of isolation of protoplasts, and then the cell wall will be formed on over this new fused cell, or the induced fusion. Induced fusion using chemical or mechanical, right? Or mechanical fusion using electrofusion, electroporator we can use for the mechanical fusion, or the chemical fusion, which is normally used using PEG or using glycol and high pH and high calcium, also sodium nitrate can also be used as a fusigen. So this methods of fusion and selection can be in 5-mark question, this separately. So the PEG induced fusion. So PEG, you might have already seen, PEG polyethylene glycol is a viscous solution. So the aim is to bring two cells together in close proximity. So when you put it in a viscous solution, two protoplasts will come together naturally and start to fuse, right? So this is the background of PEG induced fusion or the electrofusion. So using an electroporator. So when you apply a sine wave generation, a low voltage, rapidly oscillating AC current. So all those protoplasts will line line horizontally towards those two poles, positive and negative poles. And because of this, so these come in low proximity to each other, so that there will be fusing of that, right? Fusing of those protoplasts. So this, so when either this or this, right? So not all protoplasts will be fused. Only few will be fused, right? When you have many bubbles, only one or two bubbles will fuse together. Some may burst, right? Some may not fuse. So we need to select what are all those cells that are actually fused and remove all those unfused ones, right? We need to remove all the wild cells. Right, so the mechanism of protoplast fusion. So how this can happen is agglutination between the protoplast. Agglutination is coming together of those protoplasts, right? So this agglutination coming together of protoplast will lead to the fusion of plasma lemma, forming a cytoplasmic bridge, and then the rounding of the fused protoplast, and then the expansion of the cytoplasmic bridge. So this can form heterokaryon or homokaryon. Karyon is nucleus, right? Heterokaryon is two different nucleus. Homokaryon is single nucleus, right? So those fused protoplasts can have two different nucleus or a single nucleus, depending upon the genetic constitution of those protoplast. So how this can happen is, so you have two protoplasts, right? Like protoplast A and protoplast B. So protoplast A, this blue color and the green and red and pink. Okay. So when these two are fused, you have a somatic hybrid. Somatic hybrid. So you have the fusion of both the nucleus and fusion of both the cytoplasm, right? So this can be fusion of all those things. The nucleus, both the nucleus can fuse together and both the cytoplasm can fuse together. Or you can have the cytoplasm from one cell and nucleus from one cell, or a nucleus from one cell and cytoplasm from another cell. So this will be the cytoplasmic hybrids, or also called as hybrids, right? The cytoplasmic hybrids. So the protoplast fusion products we need to select. So what we need to select or interested, classic then. Yes. Check. Okay. We'll see this protoplast fusion. Next steps.